This is lecture 16 in the course Principles of Metabolism. In this lecture, we'll look at metabolism of one-carbon units and methylation reactions. So one-carbon units, what is this? Well, this is sort of the smallest building block that you can think of in metabolism. We have looked at the metabolism of acetate groups carried by acetyl-CoA, and that's a common building block. This is metabolism of methyl groups, basically. But we call it one-carbon units because these pieces of one carbon could also exist in other redox states, you might say. So it could be methyl, but it could also be methylene or formyl or something like that. So one carbon unit is a kind of umbrella term. But a lot of this is about methyl groups. So on this slide, you see some examples of metabolites that carry methyl groups and where methylation reactions are important. And as you can see, this is widespread throughout metabolism. There are a variety of examples of this. The creatine is one typical example where you have a methyl group tied to a nitrogen. Adrenaline, a particular small metabolite synthesized from tyrosine, where you also have this methyl group tied to the nitrogen. And that's a fairly common structure, that you have a methyl group attached to a nitrogen. But you also have methyl groups in other places, like in methylcytosine, in methylated DNA. You have a methyl group attached in this position on the pyrimidine ring. And also in proteins, we have a lot of methylation reactions. So lysines are typically methylated also on a nitrogen. In lipids as well, we have a lot of methylation going on. So choline, this molecule, which I think we haven't seen before, it's the head group of phospholipids. And in choline, there are 3 methyl groups sitting attached to a nitrogen. So these are some examples, and this is there are many others. So this is widespread throughout metabolism, the methylation. There are also cases where one-carbon units are used not to form methyl groups, but to just add a single carbon group, so to say, in the synthesis of larger molecules. So nucleobases and purines in particular is a good example of this. So in purines, these 2 carbons derive from one carbon units in the synthesis of these base pairs. So one carbon units are really small chemical groups. So you can imagine they have to be carried by something. We saw previously that acetyl groups and larger carbon chains, acyl groups, are carried by coenzyme A. That's a very common carrier in metabolism. So what carries the one carbon units? Well, it turns out there are 2 carriers actually. So one is S-adenosyl methionine group over here. And this is a metabolite that is basically composed of methionine, this moiety, and the methyl group is actually the methyl group that is part of methionine. And then this is attached to adenosine, the nucleoside. So that's one of the carriers, and the other one is bound to folate. So this is activated folate, tetrahydrofolate. And here the methyl group is attached to a nitrogen on this ring structure here. So this is the methyl form of one carbon units bound to folate. These are the 2 carriers. They have slightly different roles, as we will see. The folate carrier is often used in synthesis of nucleotides. And so, as you probably know, this vitamin folate is associated with cell proliferation and embryonic growth. And so folate was discovered in the 1930s by Lucy Wills as a factor that was required for pregnancies to carry to term. So there were nutrition deficiencies where people did not have enough folate, and this led to problems. So it was known as the Wills factor for a long time before the structure and the function of this was elucidated. So S-adenosylmethionine carries only methyl groups. It's the methyl group of methionine that is carried. But tetrahydrofolate can carry one carbon units in several forms. And I guess that it's really when it comes to folate that people use the term one carbon unit to really capture these different forms. So here we see the three major forms, I would say, of the one carbon unit bound to folate. So there's a methyl form, there's a methylene form, and there's a formyl version of the one carbon unit. So these are basically three different redox states. You have this one carbon, and it has more or less hydrogens and oxygens, so it's more or less reduced or oxidized. And as usual, when it comes to coenzymes, we don't really have to worry about this complicated structure. The one position where something interesting really happens is at this nitrogen and also this one. So here is where the carbon group is bound. So the methyl form is the most reduced form. So here you have a carbon carrying three hydrogens. The methylene is somewhere in between. So here the carbon is actually bridging these two nitrogens. So there's two bonds to nitrogens, which makes it more oxidized then. And then it has two hydrogens bound to it. So this is an oxidation state of 0. And then the most oxidized form is the formyl version. So here you have the carbon bound to one hydrogen, and it actually has a formaldehyde group, really. It's bound to an oxygen and a hydrogen. So folate can carry one carbon unit in these three forms. So let's look a bit more closely at the metabolism of one carbon units bound to tetrahydrofolate. So I would say that the central form really of one carbon units bound to folate is the methylene form. So this is put in the center of this picture. So this is the form in which one carbon units are synthesized. So they can derive from serine, from glycine, or from betaine. These are the major donors that give one carbon units into the system. And in most cells, serine is the major one carbon donor. It's the most common metabolite that gives rise to one carbon units. And in serine, it is this carbon on the side chain that is being extracted and bound to the tetrahydrofolate molecule. When this happens, serine is converted to glycine. So glycine has now lost this carbon. And in some cells, glycine in turn can also be used to get one more one carbon unit. So then this carbon is used, is transferred to the tetrahydrofolate, and again gives this methylene form of tetrahydrofolate. This is done by something called the glycine cleavage system, this enzyme complex. And when this happens, the carboxyl group of glycine is released as CO2. This happens mostly in the liver, and in the liver, betaine, which is the metabolite of choline, can also donate one methyl group. It actually has three methyl groups bound to a nitrogen, but it can donate one of them and also generate this metabolite. So the methylene tetrahydrofolate is kind of the central form, at least for production of one carbon units. And then, as we said, there were different redox states of this, basically. So there's methylene, there's methyl, and there's formyl. And as you would expect, these are converted back and forth by oxidoreductases. So there's a redox system going on here. For making the methyl form, there is a reduction, and as we have sort of come to expect, this is carried out by an NADPH system. You can make a reduced form. And this form can then, as we will see soon, actually feed into SAM. And so this methyl group that has been attached to THF here can then be donated to the S-adenosyl methionine carrier, and it can go on to do methylation reactions. That's one fate of carbon coming from this system. As we also said, the folate-bound 1-carbon units are used in nucleotide synthesis. And so the methylene form, CH2 form, is used to make thymidylate, the T base, a special reaction that is using this form of 1-carbon units. If the 1-carbon units are oxidized down to the formyl state, then this form of the 1-carbon units can be used to make purines. This is used in the synthesis of the purine ring. And these are the most important fates of carbon bound to folate. So as you can see, it's closely associated with nucleotide synthesis. There is also another source of these groups. So formate, the carboxylic acid, the simplest carboxylic acid, can be sort of directly bound to tetrahydrofolate. This is a ligase that requires ATP. This has been thought to be less important sort of traditionally, but in recent years there have been some studies showing that this actually occurs in some cell types. So this is potentially another source of the 1-carbon unit bound folate. The other major metabolic system is the S-adenosyl methionine, or SAM, cycle. So this is the part of 1-carbon metabolism that handles methylation. So this is the major methylation donor in mammalian cells. And you might say this is downstream of folate in some sense. So folate comes in, the carbon comes in bound to tetrahydrofolate as tetrahydrofolate methyl group. And this is donated to homocysteine, this amino acid, to form methionine. So this is also a means for methionine synthesis in some cells if homocysteine is available. And then methionine is activated, you might say, by adding the adenosine group. So here an ATP is used, and the adenosine group is attached to this sulfur atom in methionine to form S-adenosyl methionine, or SAM. This is a little unusual reaction actually, because it uses ATP, but it actually sort of releases all the phosphate groups in the form of a pyrophosphate and a single phosphate, sort of in one reaction mechanism. And it's only the adenosine group that is attached to methionine. So this molecule, SAM, is the major methyl donor in cells. So this is the methyl group that is used for all methylation reactions, really. So folate produces methyl groups bound to THF. The folate system produces that. But this is, as far as we know, this is only used as an intermediate to then give the methyl group to SAM, and SAM is the major carrier. So there's a lot of methyl transferases that methylate all sorts of things. As we saw, there are lots of small metabolites. There is DNA methylation, there is protein methylation, there is lipid methylation. So a lot of different substrates that can get methylated. So we just summarize it here as some substrate X can accept this methyl group from SAM in this reaction, and then we get the version of the SAM molecule lacking the methyl group. So this is S-adenosyl homocysteine. So now the amino acid part of this group is now homocysteine, and then the cycle is completed by cleaving off the nucleotide adenosine, and then we have homocysteine again. And then this cycle keeps turning. And this is a true cycle, you might say, because the homocysteine part stays in this cycle, it never leaves, unlike the TCA cycle as we saw before, which is kind of more like a spiral. So this is a true metabolic cycle. Now, you might wonder why you have to have two different systems. You already had the folate group carrying methyl groups, so why do you have a separate system? So one reason might be that SAM is really an excellent methyl donor because this group, where the methyl group is bound to the sulfur atom, it's kind of bound very loosely, you might say. The sulfur atom is very keen to donate this methyl group to other things. So it's a very good methyl donor. So very favorable reactions to methylate from this. Another reason might be that it is good to compartmentalize, separate the two systems, the folate system and the SAM system. So whereas folate is used a lot for nucleotide synthesis and associated with cell proliferation, SAM is used for all sorts of methylation reactions that happens in all sorts of cells. And these systems are regulated a bit differently. And so it might make sense for the cell to have two different carriers to really separate these systems. So to summarize, the one-carbon metabolism consists of these two systems, the folate system and the SAM cycle. And as we saw, the one-carbon units derive from substrate like serine and make this form of one-carbon units bound to folate, which are used for nucleotide synthesis. And then there is this bridge from the methyl formate form, which is donated to the SAM cycle and forms methionine, and it's used then for methylation of various substrates over here. Now, these systems are regulated differently, and we will just see a couple of examples of this. It's rather complicated. There are lots of feedback inhibitions and things like that you normally see in metabolism, but just look at a couple of sort of modes. So there is thought to be a kind of priority system, so that you can think of it as there are two major uses of the one carbon groups. There is the folate system that donates to nucleotides, and there is the methylation system. And if we are in a situation that we are low on one carbon units for some reason, what happens is that the cells prioritize the SAM system. At least this is the thinking right now. And so in this situation, the one carbon units are prevented from being used for nucleotide synthesis, and they are preferentially shuttled to the SAM loop. So if you have little methionine, for example, so that it is difficult to methylate, this system, this regulatory mechanism kicks in, and the one carbon units bound to folate are shuttled towards the SAM system to maintain the SAM levels and make sure that the cells have enough methyl units to be used for methylation. Because in many cell types, these methylation reactions are really vital for the cells to survive and carry out their function. In situations where you have metabolic deficiencies, like vitamin B12 deficiency, some of these regulatory systems can backfire, and this is known as the folate trap. So B12 is actually a cofactor that is required for this reaction in the SAM loop to take place. So to actually form SAM from methionine, you need B12 as a cofactor. And in some people that have metabolic deficiencies for B12 or genetic deficiencies, you can get this type of a sort of deadlock or a trap, where because you cannot form SAM, the cell interprets this as a loss of methionine or a loss of methyl groups. And then this regulatory mechanism is activated so that 1-carbon units are shuttled towards the methyl group. But because the SAM cycle is sort of broken, the SAM cycle cannot turn as normal. These folate-bound methyl groups cannot really enter the SAM cycle, and what happens then is that they accumulate in this form, or they are trapped in this form. This was a mechanism proposed some while back. It was reviewed in 1982, and it's thought to be one of the major problems in vitamin B12 deficiency. And so what you see happens is that because these two systems are connected, problems in one cycle, problems in the SAM cycle can sort of spread to problems in the folate metabolic system. And in this case, you will deplete folate-bound 1-carbon units because you have a problem in the SAM cycle. So these are two tightly connected metabolic systems that handle 1-carbon units.